Hello, everyone. Good afternoon. I hope that you have a pleasant lunch. So it's my great pleasure to part of this symposium, especially this afternoon session regarding the AMR research in Pasteur network. So today, I would like to take this opportunity to tell you the story why and how we initiate the International Research Alliance RAPID. And then I would like to talk about what we want to achieve through this alliance. So as you know, the antibiotic resistance is a big problem, and antibiotics actually have been considered as a magic bullet for bacterial infection treatment since the 1940s when penicillin was first introduced in clinics. And after that, we are heavily reliant on antibiotics. In each year, we are consuming almost 35 billion pills of the antibiotics, creating over $47 billion of antibiotic market globally. However, as we know, inappropriate use of antibiotics actually increased the antibiotic resistance and spreading all around the world. And recent Lancet paper actually reported that almost 5 million people died due to AMR, antibiotic resistance, including 1.27 million people who directly died because of the AMR. And I would like to emphasize in this manner regarding to in Asia Pacific. So among the 1.27 million deaths, actually 50% were occurred in Asia Pacific region. And then as you can see here, actually by 2050, if we don't do anything, then the death toll will increase to 4.73 million deaths in Asia, only Asia. So Asia is the most, the place will be most affected by the AMR. So this means we don't have time. This is a time to act on AMR antibiotic resistance, specifically in Asia region. So regarding these unmet needs, actually 2 years ago, 2021, the researchers from the Institut Pasteur Network Asia Pacific Hub, we got together and then did the workshop, 5 days workshop covering various topics on AMR. As you can see, we covered the diagnostics, treatment and use, immunity, personalized therapies, and technologies on AMR. So it was a very fruitful workshop. However, we didn't just stop here by just holding the workshop. We tried really hard to move forward. So after this workshop, what we did was we actually organized the committee meeting and then surveyed what kind of unmet needs researchers feel, and then what kind of common research area that we can work together. The result was on the bottom like that. So the first topic, the many researchers think it's necessary was establishment of a common database or data repository system for AMR research. I think that everybody probably in this room will agree with this part because we do have a lot of data, but if we don't share them, then it's just fragmented. If we can have big data, common database, that will facilitate the AMR research, of course. The second part the many people were interested in was strengthening of metagenomics activities in Asia region for AMR. So identifying this prioritized research topic, what we did was we actually initiated a new international R&D alliance called RAPID. So the RAPID is R&D Alliance for Preparedness of Infectious Disease. And in this RAPID, actually we had a kickoff meeting last year, And then in the kickoff meeting, we had researchers from the Institut Pasteur network in Asia Pacific, including Institut Pasteur Korea, Nha Trang in Vietnam, and Hong Kong and Cambodia, and New Caledonia and Iran. So we have all the researchers play in this rapid alliance. And in this kickoff meeting, what we also decided was, as an initial project, we actually, So we actually tried to start with the investigation of AMR in environment to applying metagenome analysis. Why environment? Because, as you know, our body has a full of microbes. Actually, it's almost 38 trillion bacteria in our body. And then these microbes actually affect our health dramatically, actually very important things, especially these days. But often we don't really realize is that approximately 10 to the 6 of particles that likely contain the microbes that are hourly shed from our body. So think about this room. So this room was full of us actually before the symposium started. If you can look at the microbiome in this auditorium, probably they will be different than right now, because right now the microbiome in this auditorium was affected by our microbes. So, you know, on your chair, you'll probably shed a lot of your microbes on the auditorium. So that's why we are quite interested in environment, especially urban environment, where the people constantly connect and interact with the environment. We think that probably in that way, a portion of our urban environment microbiome is kind of a reflection of a community level of microbiome, or including the antibiotic resistance. And this type of concept is not new. Actually, decades ago, people were thinking about the environmental antibiotic resistance as a quite important subject. And in addition to that, increasing or advancing with the metagenome or sequencing technology actually lead us to be able to look at the more deeper microbiome analysis. And then using these technology, actually nowadays people starting looking at more and more urban environment microbiome as well as antibiotic resistance. So with the similar type of research concept, actually the rapid we started a pilot project called the Toilet Project. So in this Toilet Project, what we do is that we collect the samples from the toilets in the public places like university, hospital, shopping mall, subway station, like a public transport system, parks and supermarket, etc. So we collect the samples from the toilets in these places. And in this year, starting this year, in 4 different countries, Korea, Vietnam, Hong Kong, and Cambodia. So we collect the samples quarterly. So Q1 was done this March, and then Q2 was done in June, and Q3 was just finished in the last September, and now we will collect the sample Q4. So with the Q1 through the Q4, we can complete the 1-year sampling project. So if you just think about the collection of the sample, it seems like quite easy, but actually that's not. And sample collection is quite important because it can affect the downstream of the analysis. If you just scratch the surface of the toilet, you may not get enough material, and then it will affect the downstream sequencing process. So it is very important to harmonize the protocol of the sample collection from the beginning. That's why, as an alliance, we also established a standard method for the sample collection. So we made the SOP, and even we made the sample collection tutorial video for the newcomers. So the new participants can learn how to collect the sample through this video tutorial. So once we have the sample collection and proper samples, then all the samples were subjected for the shotgun metagenome sequencing. And then with the shotgun metagenome sequencing, raw sequencing data, we actually analyze the data through our own pipeline, as well as our international collaboration, like Metasub and GOC, you probably remember from the first session. So not only analyze the data, like preliminary data, but also all the raw sequencing data are go into the common database through the NAS system. So network attached storage system. We established this data storage system because using this system, actually we can share our data freely throughout the member, wherever they are located. So what could happen is that after the sequencing, we put all the raw sequencing data, PASTA-Q file data, into the NAS system, as well as in the meantime, we also starting analyzing the data to generate at least the first layer of analysis result. All these results goes into the NAS system. And then my colleagues in different countries, actually they can access to this database storage system and download the raw PASTA-Q files, as well as analyze the data. And then they can freely use the analyzed data, but at the same time, what they could do is they could just to play with the raw sequencing data. So PASTA-Q file, they can analyze data wherever, whichever they want. So then, after that, the new analysis data goes back to the NAS system to share the result among the members. So we call it, this is the open international collaboration. By doing so, we can actually synergize the sequencing analysis result. So one team or one person's analysis, definitely not enough. Having the brains together, we can actually increase the impact of a result, and also we can analyze the data in different aspects. So that way we can extract more information from the data. So this is the kind of a schematic how we pursued our project. As an alliance, we support for the members in terms of the swap kits. So whenever members go out to collect the samples, we send the sample kits so they can, swap kits so they can sample. As well as for the sample collection, as I mentioned before, we provide the SOP as well as a video tutorial. And then also once the collaborators, they collect the samples, they can send back to Korea for the sequencing analysis, and then all the shipping costs are covered. And again, the last part is metagenome sequencing results, including raw data, are also provided to all the members. That's what Alliance provided to the members. So with this schematic, actually now I would like to show you some of the preliminary results from our Q1 sampling. As I mentioned, the Q1 sampling was done March 20th through 31st in this year. And then we performed the shotgun metagenome sequencing for Q1 samples. Actually, I forgot to mention, all the samples were collected in duplicates. So we only subjected for the sequencing one set of the samples, and the other set of the samples, we kept it for the future, actually. So this time we analyzed the data which from the 130 samples and from 4 different countries, Korea, Hong Kong, Vietnam, Cambodia. So including 8 negative controls, so we actually have the 122 samples. They were collected from various places, including actually individual household. So hospitals, subways, office tower, public toilets, university, etc. And here is our first analysis result. So if you look at the alpha diversity analysis result, it's kind of reflect the sample numbers. So you can easily imagine that the sample numbers low, you may expect a high diversity in general. But actually, it's not really significant in Shannon analysis, though. One thing I found quite unexpected one, a little bit shocked, was actually for the beta analysis. When before we analyze the data, actually we kind of expect that the microbiome samples from different countries will be clustered differently. But actually, it wasn't that case. As you can see here, so all 4 countries' samples are mingled together. We didn't know why is it at the beginning, but when we look at all the species identified from each countries, actually all of them are from either environmental microbes or human-originated microbes. It's kind of mixed together. One thing I realized that actually, as I before mentioned, we also had the individual household samples. We thought that may affect the diversity, and that it could kind of dilute the country-specific the microbes. So what we did was actually we started looking at the data place-based one, sampling site-based one. In this case, we only look at the hospital samples, so collected from the toilets in hospital. And then in this hospital samples, we have the 3 countries. And as you can see here, actually diversity, the beta analysis actually now shows that the different clustering among the different countries' sample. So that's kind of what we expected before. So again, it kind of implies that the previous entire data, when we look at it, probably because of the individual household samples could dilute the culture uniqueness. So anyway, in this case, in the hospital samples, we had the clustering among the countries' samples. And I don't know whether you can be able to see the species names. Actually, if you look at the species names, they are mostly likely originated from the human microbiome as well as opportunistic pathogens. That's what kind of what we expected result. Again, when we only select the public places, which is subways, public toilets, supermarket, and shopping mall, again, unlike the all together, actually these selected places shows the different clustering among the different countries' samples, which also, again, as we expected. And then, unlike the hospital samples, if you actually see each individual species composition, actually, we found more like environmental originated microbes, while the hospital samples are actually more likely human originated ones. So it's kind of alert that public places might be more open to the environment, so it could affect the microbiome in this way. So I don't go to the detail, but I will just show the brief result. And this is kind of another uniqueness. So among the collection site, actually university, I specifically select the university because unlike just the public places, we don't know exact users. It could be children, it could be elders, we don't know exactly. But if we specify the sampling site, like a university, you can kind of imagine that the users could be certain age ranges, and then likely healthier. So in that, in this assumption, actually, if you look at the alpha analysis result, actually, you will be able to see the more clear clustering between the different countries. And I think that we haven't really into the deeper analysis, so what kind of, implication this result has. But one thing is that by doing this type of exercise, what we learned is that we need to actually decide what we want to see and then specify the samples to be able to see what we want to see. Like in a university case, we kind of narrow down the age range things. So obviously, the most important part is the antimicrobial resistant gene distribution. So among the 122 samples, that's the heat map chart. So what we found is that in general, actually, the resistant genes for the aminoglycoside and beta-lactams, MRS, and tetracyclines are quite prevalent throughout the entire different countries. And by curious, so I actually look at the antibiotic consumption rate for each country. So it's, in the case of Hong Kong, it's the data from the 2021. Actually, the beta-lactam, tetracycline, MRS, and quinolones, fluoroquinolones, are the kind of top sell prescribed antibiotics. And Korea and Vietnam is a kind of a similar way. So it's not really a perfect match, but still you can see that there might be a strong connection between the antibiotic resistance prevalence with the antibiotic prescription rate. Another thing that we actually look at was, in this chart, what I want to show you is that actually still is the antibiotic resistant gene presence, but each color has the different genes, which encodes the resistant genes against one particular class of antibiotics. So in this case, probably you don't see a lot much, but what I want to show you is that each gene, actually these type aminoglycoside resistant genes, so here and here, you see the white dots are spread across over the all countries, all the samples. So these are the most popular genes for the aminoglycoside resistant genes. And for the other class of antibiotics, a similar way. So that means that actually it implies that resistant genes are not really, these genes, especially these genes, are quite prevalent over the all cross over the countries. But again, you will be able to see specific genes that are quite present in particular countries, but not other countries. So maybe these genes, we need to look into more to see whether these genes actually has an impact in clinics. So, but we haven't go into that far yet, but we are kind of still scratching the first layer of analysis here. So, in terms of just the antibiotic resistant genes, not globally, but again, when we look at the overall, the diversity level, it's kind of similar to the microbe taxonomic analysis result. And I'm saying that because if you look at this one, actually, if you just selected the hospital samples, the big graph, this one is the alpha analysis result based on the ARG, antimicrobial resistant genes. And then this small one, you have already seen from the taxonomic analysis. Can you see the resemblance? So it's quite similar. And then same thing goes the beta analysis as well. And if you go to the actually public places, again, the patterns, as same here from the ARG, alpha analysis result, and taxonomic analysis result. To me, it kind of implies that somehow these antibiotic resistance genes might be associated with the taxonomy. So
sorry, Dr. Soojin.
Yes.
It's time, but please accelerate.
Yes, I will do that. Okay, so that's why we are doing currently this way. So we are trying to make the co-occurrence analysis to see what kind of resistance genes appear the same time with the certain microbes, but it's in progress. I hope that I can show you this result in near future. And then with a lot of limitation, and also our study is just initiated, but we believe that our project can have the impact, not only the basic research, but at the same time public health, by doing extend the knowledge of potential pathogens and antibiotic resistance mechanisms, as well as establishing of big data, because big data that we need for the future AMR research. And also it could be an indicator whether what kind of antibiotic resistance genes are present in community, not patient, in community, that could actually show us what's coming. And also it could be used as an evaluation method if something happens and then we do the measuring, and then after the measuring, by doing this type of analysis, so we can see whether measuring works well or not. So the last one, actually, this is the kind of what I want to show you. So go together. We believe that this is the very important one. With the Rapid Alliance, currently we do the research area, antibiotic resistance, and actually today I only show you the environmental AMR surveillance based on the metagenome analysis, but as a sub-project, we also have the small project in the Rapid to identify the new antibacterial agent from the Asia-originated natural product. So with that, our collaboration work actually introduced in the UN General Assembly meeting early this year as an example of international collaboration work. So we hope that we can move forward strongly. So with that, I'd like to thank everyone in the Alliance. So thank you for your attention.